All right. Well, thank you, Professor Scher, for that wonderful introduction. It's a pleasure to be here. And we'll get started. So, what do I do? As a biologist, I'm really a bad biologist when it comes to staying in my lane. I'm not good at that. I like to study lots of different things. So what I've typically done is I've integrated approaches in evolution, ecology, psychology, biology, and I basically put that all together in a big pot, stir it up, and make things happen out of it, right? We ask big questions, and we try to figure out how do these animals evolve such interesting and dynamic adaptations that help them survive and reproduce in a fairly challenging world, right? So that's what I do. Now, as part of that question, as part of these types of questions, one of the things I like to do is think about it in two different ways, any question. So the first question you might have is, how does this thing come to be in biology? That's an evolutionary question. Sometimes we call that the ultimate questions about biology. But sometimes in biology, we want to know how the machine just works. What are the parts of the biological entity in front of us that make all of these cool things happen? And that's a proximate question. All right, and so sometimes biologists think about the evolution of the system, and sometimes biologists think about the mechanics of the system and do both things. All right, so I like to do both of that. We work in wild settings, we work in zoological settings, we put that together to find the best way to answer these big important questions. And then finally, and one of the more important things that I think that our lab is working on is thinking about all of this stuff and how it applies to helping conserve these animals. We are undergoing the 6th mass extinction right now. So just to give you a sense of my study subject, I was born around 1979. Okay, in 1979, there were 80% more bottlenose dolphins on this planet than there are today, and that's not even one of the endangered marine mammals. So everything that we focus on, it's not going to mean much if these animals aren't around for us to continue studying them. So conservation is a huge component of what we do. All right, so what am I going to talk to you about today? So we'll start with some big, heady questions about long-term social recognition and what it means for the evolution of intelligence. We'll talk about new sensory capabilities that we've discovered in dolphins and what they can do with them. And we will focus on how we're conserving these marine mammals using new high-tech drones that we're developing. All right, so first question I would have, what makes an animal smart? What do you think? What do you think? Social interaction. What else? Ability to adapt. Flexible adaptation. Cool. What else? Size of the brain. Size of the brain. Some big animals that I studied with some very big brains. Problem-solving capability.
The ability to look at themselves in the mirror and recognize that they're themselves.
Ooh, mere self-recognition. How existential. All right, differentiation between different things, right? What is this? Is this object something that is useful to me biologically? Is this something that, for example, some dolphins are even known to use tools and can recognize their own tools versus another dolphin's separate tools. They basically take little shells, put them on their nose, and dig through the sand looking for flatfish. So there's object recognition and tool use. So there's a lot of different ways to answer this question, right? A lot of different levels to answer this question at. You might have certain traits that you might think, oh, that makes that animal really smart. You know, for example, New Caledonian crows, they want to drink some water in a bottle. So what they do is they put their beak into the hole opening. And as they put their beak in, they can't reach the water. So they go and they get rocks. And they put the rocks in the bottle where the water is, and it causes the water level to rise, Archimedes. And then they drink the water. That's pretty smart, right? How many of us would have thought to do that? That's a cool trick. So those are things that we think of as smart. They're flexible, they're adaptive, they allow an organism to basically look at the world around it, solve problems. All right? But let's think about that question in a broader sense. What makes an animal smart in an evolutionary sense? All right, how do we go from an organism with less cognitive flexibility to an organism with a lot of cognitive flexibility? And one of the arguments, and I heard this mentioned, the idea about sociality. And so authors Byrne and Whiten came up with this idea that your social system can predict your intelligence. The animals that tended to be more intelligent tended to be more social. Octopus are weird. They're different. We'll put them over there for now. There was a subset of that hypothesis, that social intelligence hypothesis, that was determined Machiavellian intelligence. Who's ever heard of Machiavelli? Yeah, Machiavelli. What is he famous for? What quote? What's the quote?
Ends justify the means.
Yep, and the other one, might makes right, right? That general sense of, it's almost Darwinistic, isn't it? It's power dictates success in some of these ways. And so Machiavelli was very much thinking about these terms and these ideas. And when you're an evolutionary biologist, you tend to think of out-surviving, out-reproducing your competition. And so Byrne Whiten, very much in the primate world, start thinking about social intelligence not as just very basic kind of just big social systems equals big brains. They were thinking about it at the mechanistic level. They were saying, wait a second. If I lie, if I cheat, if I deceive, and I do so better than anybody else, I will produce more offspring in the next generation. It's very cynical. Sounds like Survivor in biology, right? Outwit, outsurvive, outcompete. Well, that works pretty well for primates. We do know that chimpanzees' society is a lot like that. We see some evidence for some of that type of social manipulation in bonobos and other great apes. We don't see it a lot, though, in other species that we think of as big-brained, smart animals. Elephants are not necessarily big on deception. Dolphins, not necessarily known for deception. So what is it? What's going on here? And maybe is there a simpler explanation, one a little less cynical, that kind of explains a broader evolutionary intelligence? And so what we did is I looked around in a lot of the literature about what we knew about how well animals could remember other animals, because that really steps everything back a bit. Take it back a bit and say, okay, what is the basic intelligence that a social animal would need to have? And it's just keeping track of everybody. They don't have Facebook, they don't have social media to have all their friends on a list for them. They've got to keep track of it all in their head. And so maybe that's the driver of intelligence. All right? And again, that's something that whether you lie, cheat, steal, or deceive or don't, you still need to do to be smart. All right? It may drive other intelligences. And so you see a huge variety of some of these kind of lengths of time, the different modalities, the different sensory systems they use. And one thing that really popped out here, with the exception of one anecdotal example in elephants, one of the tricks that really defines an animal's ability to evolve intelligence when it comes to just keeping track of everybody is who you keep track of. You keep track of your kin, and you keep track of your non-kin as humans, don't you? Picture your best friend right now. Okay, picture a family member. You did that, right? Anyone struggle with that? No. You can do that. You can do that equally well. It turns out the animals with the most complex social systems, the fission-fusion animals, animals that live in complex societies like we do, where you break up your social group, form different social groups, and kind of mix and match a little bit. Those are the animals that can remember kin and non-kin equally well. The animals that don't do that, don't have that complex social system, usually can only identify kin. And it's usually somewhat cheating. What they do is they sniff themselves. And then they sniff their neighbor. And if their neighbor smells like they do, I treat you as familiar. I treat you as kin. It's called self-referent phenotype matching, or the armpit effect. And it's not really that they're remembering anybody, they're just remembering they're just basically noting who smells like they do. So there's the trick, right? There's the trick that a lot of animals use. It's very adaptive, but it doesn't require a lot of cognitive horsepower. All right, so here we go. We've got something we can look at across multiple species to see if it's something as basic as just keeping track of everybody that defines intelligence. All right, so let's talk about bottlenose dolphins for a moment. This is my study species. Generally, we think of bottlenose dolphins as living about 20 years, but there's a huge range in that age, okay? Because a lot of calves don't make it past the first year because it's a lot to ask a mammal to be born in the ocean. So that can get a little rough. So we do have some maximum ages of males living somewhere above 40 years at a maximum, and females living above 50 years if they're so lucky. Now, bottlenose dolphins do not live in pods. Okay, pod implies that it's like peas in a pod, and the peas don't separate from the pod, right? Dolphins, again, live in a fission-fusion society. They break up that group, they go to different groups. It's like having your sports team friends and your school friends and your family. These are different social groups that may have some overlap, but don't necessarily have to. And so because peas don't break up out of peas out of a pod, we don't say that dolphins are pod members. What they are groups. And those groups can aggregate into larger groups of over 100 different individuals. And they do that for two reasons. One, a dolphin cannot catch a fish one-on-one easily. Okay? Fish can turn on a dime. A dolphin's like a Mack truck. It keeps going. So what do? They work together to corral the fish into each other's mouths. That's why they're so cooperative. That's why they're so social. Okay? The other thing is cooperation and predation defense. One of the big questions everybody asks me is who wins in a fight, a dolphin or a shark? I say, well, one-on-one, it's a good question. But the dolphin is almost never one-on-one. The dolphin's got his gang with him. And that's why the dolphin wins. Okay? So, cooperation in defense, and especially defending the calves. Everybody works to help defend the calves. Now, how these groups get together, it's not just random. It's not just randomly who associates with who. It's like high school. Cool kids hanging out with the cool kids, less cool kids hanging out with the less cool kids, and then there was my group that was just the sci-fi nerds, just off to the side. And that's a little bit what dolphins do. Their social status influences their patterns of association. So there's cool dolphins and less cool dolphins. It happens. Now, dolphins are also notorious for their vocalizations, and many people ask me, do dolphins have a language? And the answer to that is, we don't know. What we do know, though, is that dolphins have signature whistles, which are individually specific contours, different individually specific calls that they give when they're separated from their main groups. And so the way a dolphin lives is if it separates from its group, and it says, hey, wait a sec, where's everybody? They'll start saying their name. Jason, 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 Jason. And that's how the other dolphins basically play Marco Polo to find each other, because sometimes it's hard to see in the water. Now, when a calf is just about to be born, the mother starts whistling her signature whistle over and over and over again. And that's because the calf has to learn in the womb where to go to find mom. And so she's basically, we call it a recall, calls the calf back to mom. All right, and she'll do that even after the calf is born. So you can actually time when the mom's going to have their calf based upon how many signature whistles the mom gives. Now, there is a point where the dolphin has to swim at depth, and at that point, the frequency of their whistle goes up. It's like humans on helium. And so because their voice changes depending on how deep they are in the water column, they do not have voices. So they use these individual shapes of the whistle, and I'll show you that in a moment, to basically act as the signature whistle. Okay, so they each innovate their own signature whistle. They name themselves in that first year of life. But what they'll do is they'll listen to dolphins that swim up and hang out with the pod for a little bit and then swim away maybe a day or two, because those are the dolphins that have the most favored elements to be part of their signature whistle. That prevents everybody from being named Bob. All right. So here's some signature whistles. This is a dolphin named Allison. She has 2 S's. This is her sister, Nolani. And this is their mom, Tupiko. You've just heard 3 dolphins give their names. Now, Tupiko's signature whistle is unique. It's an upsweep and a series of hooks. All right, so they look like little hook shapes, right? And the rules for Tupiko are different than any other dolphin. If she gives an upsweep 1 hook, that's her signature whistle. If she gives an upsweep 5 hooks, that's her signature whistle. Why she varies it, we don't know. But those are the rules for Tupiko, and every dolphin has to learn. That's how Tupiko does her voice. That's how she does her name. Nolani is always that single flat tone there. Sometimes she'll repeat it over and over again to get anyone's attention. But basically, it's a single flat line here. And Allison always does 2 S's. So every dolphin comes up with the rules for their name, and then that's what gets communicated to the rest of the group. That's how they know who each other is. Now, here's my question. On an evolutionary sense, do dolphins remember signature whistles for long periods of time, providing evidence for that social intelligence hypothesis? And do they do so as a means of possibly explaining the evolution of their cognitive abilities, all the other cool stuff they can do? And so that's really what we're asking. And to do this, I went around the country and Bermuda to basically play telephone between dolphins and their signature whistles. So record their signature whistle at one place, play it back to a former social partner that hadn't seen them in years. Now, of course, you can do this a couple different ways, but one of the nicer ways to do it, if you've not lived a very, very long time, at the time I had done this, I hadn't lived a very long time, you can also go to the Woods Hole Oceanographic Institution and get a bunch of other whistles from dolphins who had long since departed, shuffled off the mortal coil, if you know what I mean. So, what happens? You basically take those whistles, you get 53 subjects together across all those facilities, and then you add in all of those whistles you get from Woods Hole of dolphins from the far, far past, and get a bunch of signature whistles to play back to dolphins ages 1 to 46 years of age, with about 40% born in the wild and 60% born under human care. The data I'm about to show you tested a lot of dolphins. 1,300 playbacks over 5 years, testing 266 different dolphin relationships. So this is a pretty comprehensive data set. Now you might ask yourself, how do you talk to dolphins? Well, carefully. First thing you do is you get an underwater speaker and you put that in an area they cannot access. All right? Once you do that, the dolphin basically gets itself, usually as it's kind of getting into its half-brain sleep thing, it gets into a circle swim pattern. When it gets into that circle swim pattern, you present a playback as the dolphin's head crosses this imaginary line, in which case now you look and see how does the dolphin interact with the speaker. A level 0 is they didn't care what I played. No response. Level 1, you get a little head turn. Dolphin swims by, turns their head, and keeps going. Level 2, the dolphin loops over, investigates the speaker, but then kind of keeps going. Level 3 is when the dolphin hangs out at your speaker for a period of time. And a level 4 is where the dolphin would make a forceful contact with the gate or start speed swimming. This only ever happened once, and because it happened, we were able to move the 2 dolphins to live with each other because it told us, hey, these guys really need to be with each other. And it was a really nice way of assessing that type of information. You couldn't have gotten any other way. So it was good that we did that, so we knew. All right, so here is a dolphin response. The first sound you hear is my playback. Everything else you're hearing is the dolphin responding to its familiar partner. Now, to provide context for that social interaction, the dolphin that you're looking at there, his name is Kai, he knew the dolphin we played along with the other 2 dolphins that swam in there. They knew that guy for about 5 years ago. They hadn't seen him in 5 years. The male they were responding to was the dominant male, Hastings. And so all of that posturing that you saw was them being all tough and mighty to an animal behind a gate. If that dolphin had been in the pool with them, their answers would have been very different to that playback. Okay, so let's take a look at how I would do a typical playback session. First thing I want to do is I'll play a dolphin that they do not know. All right? And I keep playing dolphins they don't know until they stop responding. Eventually they get bored of this. Okay? Then I play a test whistle. And let's say it's a familiar test whistle in this case. All right? We look at the response. Then we play another unfamiliar whistle, and we keep playing unfamiliars again until they stop responding. And let's say they stop responding in this case, we would then play a matched unfamiliar test whistle, matched by age and sex to our test whistle here. So the comparisons you're about to see are between this response and this response. And sometimes this was over here, and sometimes this was over here. So we randomized the order of these playbacks. All right, so let's take a look at what I was interested in. First thing I'm interested in is sex differences. Are males better at remembering than females? Or the other way around? Age differences. At what age can dolphins do this thing? Remember another dolphin signature whistle. That kin-non-kin question. Can you remember kin as well as you remember your non-kin? That seems to be a very important thing in animal minds. All right? I'm also interested in how long dolphins can remember each other after they're separated. That's the big question. But I also want to know how long dolphins can be together to form a memory. All right? Do they have to be together for a really long time before they form the memory, or can they be together for a really short time to form a long-term memory? And so what you're seeing here is evidence for the longest memory ever seen outside of humans. Dolphins can remember another dolphin for more than 20 years. And in fact, we did a version of this study as an anecdote to a dolphin we found had a 40-year separation from another dolphin. And we did the playbacks, we did the controls, we did all of it. And when we played this male dolphin to this 50-year-old female, she hadn't seen this guy since they were 3 years old together, just about. She slaps the speaker with her tail. I don't know what he did, but a 43-year-old grudge is pretty impressive. So, 0 to 5 years, 5 to 10 years, 10 to 15 years, 15 to 20 years, all of these dolphins are responding significantly more to the familiar than the white unfamiliar bar. All right? We do some statistical modeling with that. We look at what's called an interaction effect. An interaction effect would tell us perhaps what the upper limit of that memory could be. The interaction is non-significant. What does that mean? It means I can't tell you what the upper limit of their memory is. It could be very well through their entire lives. This is unbelievable memory. This is an unbelievable ability. All right? Before you say, okay, wait a sec, what about elephants? We haven't tested this in elephants. We don't know. So this is one of the only systematic studies that's ever been done to address long-term memory this way. All right, let's look at some of those other variables. So how long does a dolphin have to be associated with another dolphin before they can form a memory? Well, what I did is I just took that familiar response score and that unfamiliar response score, and I just subtract them from each other just so I can put it on one axis right here. So that's what that response difference is now. Okay? It's just a measure of familiar minus unfamiliar. If dolphins need to be around each other for a long period of time to remember each other, all those little dots would fit into this box. They do not do that. They fit in this box, meaning this is non-significant, meaning dolphins don't have to be around each other very long to form a memory. That's pretty cool. That means they're forming the memories very quickly. What about age differences? Well, what did I say about calves? Do you remember how they form their signature whistle? Who do they listen for? Yeah, it's a dolphin that just comes in, hangs out with the pod, and leaves, right? Dolphins that tangentially associate with the group. So here I come to their home, and I play 130 unknown dolphins to them in the course of a few weeks. That's a lot to base a signature whistle off of, isn't it? They're basically listening to all those whistles saying, hey, that's a great idea for my name. And so calves are pretty undiscriminating, or less discriminating than the adults are, because they're really focused on listening to all those unfamiliar whistles I'm playing. And they're using it to make their own signature whistles. Oops. And you can see that that's significantly different than the adults. What about sex differences? Well, turns out males are also somewhat listening to the unfamiliar whistles I'm playing in the pool, because they're males and they're concerned about who's might be kind of moving in on their territory. So there's a little bit of a trending difference here with the males with a lower difference in response because they were more focused on those unfamiliar whistles. But here is a non-significant bar that means a lot. This shows that kin and non-kin are both recognized the same way you can recognize your best friend, or you can recognize a parent or a family member. So they're not just doing that smell yourself armpit trick. So this is the longest memory ever seen outside of human beings. Independent of exposure, they can be around each other for only a short period of time and still form lifelong memories. There are some trending sex differences that are interesting. The calves are far less discriminating, and kin and non-kin are both recognized. So what's the benefit to this, right? I can tell you this trick. Dolphins can recognize each other about a mile out. That's how far their whistles can travel. So imagine for a moment that you can get a sense that there's another dolphin out there that you know a mile away from you. Really good way to avoid dolphins that kicked your butt 5 years ago. Also, really good at determining where your alliance partners are, because dolphins form alliances. Okay, so we've got that going. Really good way to identify previous successful mates. There you go. And similarly, as you can see here, maybe it's a good way to recognize who your kin are over long periods of time so you can avoid that. Yeah. So I'm excited. I come up with this. This research goes all over the place. It's in National Geographic. I'm excited. Oh, yeah, we got it, right? And my colleagues come back and say, well, you did a really good job showing that dolphins remember each other's whistles. You haven't shown that dolphins remember each other. And I'm like, what? What do you mean? Of course they remember each other. That's their signature whistles. No. You showed that they can remember the whistle. You didn't show they remember the individual. i.e., you didn't show that the whistles were representational. So if I say, Professor Scheer, you guys can picture who that is in your minds, right? I have to show that with dolphins before I can say that they're remembering each other. To make this make a little more sense, are dolphins remembering each other, or are they remembering each other's whistles? Like, for example, have you ever heard a song on the radio, and you know the tune, but you don't know who sang it, and you don't remember the name of the song? That's what I've got to get at here. Whew, that's heady. How do I get inside the mind of a dolphin to answer that question? Well, I looked into the research on how do you determine representational signaling. And while dolphins don't have voices, horses do. And there were some colleagues that did some really good work with horses. And what they would do is they would have horses in a pasture. And what they do is they take the horse, we're going to lead you into the barn, horse, and they lead the horse into the barn, and they have a playback speaker sitting right in the barn. And then they play a sound of a horse call. And if the horse call matches the horse they brought in the barn, the other horses in the pasture don't care. They don't look up, they don't do anything. If, however, you take the horse, you lead it into the barn, and you play the wrong call, a different horse, all the horses in the pasture do this. It's weird. It's called a violation of expectation paradigm. I was like, okay, maybe we can do that with dolphins. I'll get a giant high-definition screen, because I can't move dolphins like horses through barns, but I'll get a high-definition screen, we'll video the dolphins, we'll waterproof the screen, we'll put it into the pool, we'll play different sounds, and we'll compare it, just like they compared the sounds of horses to the actual horse. Problem is, screens are heavy, expensive, and huge. And doing that would have involved hydraulics, and trying to get across different facilities with that would have been a nightmare. And so I have to deal with this question. How do I show representation in signature whistles? And so one of the things that I had recalled from my PhD work was reading an article in a book by a gentleman named Ken Norris, who was one of the foundational scientists of marine mammals. And he noted, as an anecdote in his book, and this is why books are really good, because it's a place for scientists to put weird stuff that they found that they can't necessarily publish in peer review. And he put in a little story about how when he would be going at slow speed at the front of his boat, he'd look over the edge of the boat and he'd watch the dolphins. And the dolphins were peeing and pooping, and then the other dolphin would swim up behind and just open its mouth and taste it. Ew. But that happened, and he noted that. And I said, there's got to be something to that. What if we could see if they could identify each other using urine cues? Take those urine cues and see if they've got social recognition capabilities. It wouldn't be the first for an animal to kind of use chemical cues to identify. What does your dog do if it comes up to a fire hydrant? It's checking their email. Now, dolphins do not have the ability to smell. They've lost their olfactory bulb over evolution. Their nose is their blowhole. So they can't smell, but they can taste. And so we wanted to see if dolphins could recognize each other with urine cues, and if they could, we want to use those urine cues in that cross-modal experiment. So here are the two experiments. One, to assess familiarity tests. Get urine from familiar dolphins and unfamiliar dolphins and see if they can recognize it. Two, to pair whistles with sound playbacks, just in that horse experiment, and see if they can use those whistles to identify a representational understanding of another dolphin's whistle. So you may be asking yourself, and you should be at this point, how do you get urine from a dolphin? Again, carefully. First thing you do is you ultrasound, because it's not nice to ask for the dolphin to give you urine if it doesn't have any. So you determine the bladder is full. If you do, you apply a little bit of pressure to the dolphin, and the dolphin will start to urinate. In which case, you collect, again carefully. Once you collect the urine, you usually have about 60 milliliters of, in this case, playback material. All right, because we're going to present that to the dolphin in a urine recognition test. All right, now, generally speaking, I can say that the dolphins were very willing to participate in this. To this day, the subjects of this study, if you flip them upside down, just like you saw in that picture, they will pee all over people. So they're still used to that experiment. They got so much fish for doing this. All right, so we take the urine, we put it in a little cup at the end of a pole, and the dolphins basically follow us around, all the way around a dock area. We put the urine in the water, they taste it. The variable I'm looking for here is how long they keep their mouths open sampling the urine. If they have their mouths open longer for familiar versus unfamiliar, then we know we got something, right? And that's exactly what we see. In fact, dolphins are able to recognize familiar urine at a very high rate compared to the unfamiliar urine, all right? Upwards of, they keep their mouths open about 7 seconds for familiar urine and only about 2.38 seconds for unfamiliar urine. So those dolphins just told me they can recognize each other through tasting urine cues. Not only that, and here's the fun part, they did this thing. So this is the signature whistle of a dolphin whose urine I just presented to the other dolphins. This is what they whistled back to me when I gave them the urine. They were labeling the urine with the dolphin's signature whistle. They were telling me which dolphin they were tasting. Not just once. Here's another example, and it happened about 6 other times. Yeah. So there's a dolphin telling you whose urine it is. That's pretty conclusive. Here's matches and mismatches. So take a look at the mismatch first. There's the urine, and here's the signature whistle. There it is. done there. So what we saw there was a little different than what we expected. If we were going to look at what the horses were doing, the horses spent more time during the mismatches than the matches. So what's happening here? Turns out the data show that the dolphins are more focused on matches than mismatches. And we were puzzled because, again, they've shown that they can tell the difference between a match and a mismatch. That's all we needed them to do. But why was the match more salient to them than the mismatch? It turns out horses are weird. Everybody else kind of focuses more on matches than mismatches. If you do this with human children, with pictures of family members and voices, human children will spend more time paying attention to matches than they do mismatches. So dolphins are acting a little more like people than horses. So this is actually kind of good. It shows what we were looking for, and this time it's how long the dolphins are attending toward that speaker. All right, they're responding more to matches than mismatches, which is telling us that they're assigning the whistles correctly to the individuals who own them. Furthermore, we're getting a lot of acoustic data here. So dolphins are responding acoustically with reply whistles to urine more than they do water, which says urine is a social signal. All right, we see a little bit of a difference here in the familiar versus the unfamiliar, so they're more likely to give a reply whistle to a familiar individual. No differences in the matches and mismatches. And then dolphins are more likely to investigate the area with echolocation if it's a urine cue versus just a water cue, a water control. No differences here on familiar, unfamiliar, or match or mismatch. So that's kind of cool. It tells us that the urine is very much perceived as a social signal. So why is this cool? Well, first of all, bottlenose dolphins are the first vertebrate species known to use gustatory-only social recognition. No other animal can do this that we know of at this time. With a backbone, no. Mantis shrimp can kind of do it, but their chemosenses are a little different than a vertebrate. They're also the first animal that we've ever shown that uses names the way we do. They innovate, they name themselves. Those names are essentially arbitrary, the way our words are arbitrary. Therefore, the signature whistles they develop, once they take on that representational aspect, they become exactly the same as our names. So dolphins are communicating with names the way we might. Now, there's a couple things about dolphin anatomy that's kind of unique here. First of all, dolphins possess muscular baskets in their kidneys, which we've never known what the function of these things were. It turns out what they might be doing is helping to filter compounds into the urine that are used for individual social recognition. We also think that you need a long-chain molecule to be read a little bit like a barcode in order for the dolphin to identify another dolphin. And very few molecules will get you that the way that a lipid will. So we looked at their taste receptor genes, and it turns out while they've lost a lot of taste receptor genes for things like umami and some of the sours are a little off, they still have what's called a CD36 lipid taste receptor. You have these too, by the way. We all do. And it's what tells us we've had enough food to eat. It's part of our appetitive response. Turns out that these dolphins may have a very elaborate version of a CD36 lipid receptor that allows them to recognize individual compounds in urine. The problem with this is that we can't study this in humans easily, because this is why this is a cartoon of that CD36 lipid antenna off of a taste bud. The reason we have a cartoon is because it's a very hard thing to image. It's very small, it's very dainty on top of those taste buds, hard to excise. All right? But if dolphins have a bigger version of this, one that's more easily studied, that could help us. So, here's our next steps. First of all, we want to determine what are the proximate chemical cues these dolphins are using for individual gustatory social recognition, because this is really cool. The next thing we want to know is what is the role of that CD36 lipid taste receptor, and decide if there are implications for human physiology so that we might be able to help the more than people who lack that CD36 lipid taste receptor because they are at a higher incidence of possibly being obese if they have that lost gene. So this could have a lot of implications for human health. So I'm going to shift focus just a little bit here quickly to talk about some of our conservation work. Now this is some folks in Florida doing a health assessment on a dolphin. And this poor dolphin, in this moment, is getting blood taken, getting a full health exam, and then he will be released out into the wild. But he is nice and strong, and he is capable of surviving this intervention. These are the dolphins that live fairly close to here in Galveston. They do not look healthy and strong. They are not. So if you want to do a health assessment on a dolphin in Galveston, you are not going to drag them up on a boat. Okay? This is what the dolphins near us live by. All right? This is rough. And we have a lot of that. Okay? Texas Marine Mammal Stranding Network does a lot to help try to save some of these dolphins, but there's a lot of devastation going on because of the things that humans do to these animals, especially near here. So we need to do those types of health assessments in dolphins, but we need to do it in a way that does not involve handling the animal. So how do we do it? Well, we know we've got some folks that fly drones awfully close to these big whales and collect hormone samples and health data using drones with petri dishes all over them, which is great. Except these guys don't respond well to those drones. We've got data to show you that they don't. So how do we collect this type of data from dolphins with a much smaller blow field and who are much more skittish around drones? We need to build a new drone. And so we have, over the course of the last 5 years, been developing a new drone system that allows us to collect hormone samples from small dolphins, from small whales, like bottlenose dolphins, in a way that does not harm them or hurt them. So I want to thank my collaborators first on this, a lot of our engineer teams and our biology teams, Jamie Jacob, Rick Gaeta, Aaron Alexander is going to show you I'm going to show you some of his modeling work. Haley O'Brien, who's done some of those beautiful images you're going to see of micro CT scans. So I want to give them some credit. I also want to give credit to our graduate students who've done a lot of work here, getting PhDs and master's degrees on this project, some of which are in my lab. I also want to thank the undergraduates who've worked with us on this project before I continue on, give them some credit. So first thing we did is we did some playbacks with dolphins of different drone sounds. And we did this kind of about 30 feet above their heads with an air speaker. When we did the playbacks, we found that one of the most commonly used drones in cetacean research is the one that they're most afraid of. Oops. These bars indicate how long the dolphins spend with their heads underwater. And they spend quite a bit of time with their heads underwater when you fly a Phantom anywhere near them. So, M600, this big, scary drone, they actually don't respond that much to it. Fixed-wing drones are pretty quiet. They kind of glide a little more. Don't respond much to it. And this was our control tone, just playing a whistle. So, tells us a lot about what drone not to use. We then decided to bring the real drones out and start testing them. So we used some commonly used platforms. And this is the Inspire 2. This is a thermal Mavic 2. And this is that Mini 2. We did the playbacks, and we looked for this. We start at 300 feet and descend down by 10 feet every 10 seconds. When the dolphins look up, we know we've got caught. All right. Oh, now we're caught. And Inspire gets caught a lot more. They're pretty good at detecting the Inspire. So much so, in fact, and here's what's interesting, they can see it at really well at like 290 feet. All right. And then at this point, at about 170, they stop looking up at it because they're hiding under the water. And we see that here in these data. They start hiding. So don't use that one. We're now starting to test a few other drones. Avatas, this is a waterproof drone that you can land in the water, new minis, and then we'll talk a little bit about this one here. That's a special drone. The other thing we needed to do, not only to deal with acoustics, we also have to know what they can see, because we're going to fly within about 2 feet of their head. And we got to know what they can see around them, right? To collect that blow sample. So we had to figure out what their visual field looks like. This is us training the dolphin to whistle when it sees a little light turn on. And then we put lights all around this little dome and would turn the lights on and off. And if they whistled, we knew that they could see the light. If they didn't whistle, they couldn't see the light. All right, so here's that training. So they're rewarding them for sitting nicely and say thank you.
Good boy.
Good job. Good job.
Yeah, you're doing a great job. Nice one, Coop. Good boy. turn.
Yeah, that light will turn on.
Yay!
There it
good
goes, turns on,
job.
and then he whistles.
Nice one, buddy.
So this is him being trained to do that. He got good at that. So good, in fact, that we were able to get the visual field of the bottlenose dolphin. Red is where they can't see, green is where they can. That tells us exactly where our drones can go. All right? So we want to stay in the red area. All right, we also had to build a chuff simulator because we couldn't take dolphins all over with us to Oklahoma and Texas, so we had to basically rebuild their respiratory system. We did that by high-speed photography. There's my hair with my long hair days. And that's me getting snot in my face. That's how high the snot goes that we want for the health assessment, right? So we did a lot of that, and when we looked at the data in what's called particle image velocimetry, we're able to see where every little speck of snot and water goes when they breathe. And that allows us to build our simulator. This is that simulator. This machine is actually very large. It's bigger than my arm wingspan. So you can give you a sense of how big we have to design it to match what biology can do with evolution. That's the system itself. This is how it basically uses a piston pump system to blow air through here and up through a nozzle. Now, that was our chuff sim progression, and then we looked at that and I said, well, that's fine, but it doesn't look like a dolphin's blow. We were missing the nasal passage. So that's where we had to incorporate Haley's work, scanning one of our dolphin heads, to get essentially the nasal passage down. This is the highest resolution CT scan ever conducted on a dolphin, ever. Every pixel to this is the width of a human hair. That's how accurate our system was. From there, we were able to derive out the nasal passage and basically put it into 3D CAD design. We took that, basically made something we could do a 3D printing out of, and we made a 3D printing of the nasal passage of a dolphin with a flange on it that we were able to plug into chuff sim. Once we did that, we were able to test out the system with what's called a respirometer from Andreas Fallman. This is the same unit he uses on real dolphins. He came and tested it on our system. We were able to determine that our numbers are right in the middle of an actual dolphin's numbers for their breath capability. So we're very happy about that. And our system works really, really well up to about 100 PSI. Keeps nice and consistent numbers. So we know that each breath our fake dolphin takes is pretty accurate. This allowed us to test numerous drone types. And as we predicted, the second you try to fly any of these rotary drones anywhere near the chuff sim, look what it does to all that blow. Blows it away. Not helpful. We tried airplane-looking drones, and that's all we got. 2 little specks. That isn't helpful. We need to get more snot than that. So we built PHASM. Whoop, come back. There it is, PHASM. This is a modified VTOL system. It means it takes off in the air like this, and it has variable speed, so it's an airplane that you can control the speed of. All right? And the cool thing is, it's the best of both worlds. Once it takes off, it's flown by a pilot who sees it through VR. So that as the pilot moves their head, they see exactly where the animal is. The system also has a siphon that we designed underneath it to pull the snot in to the collecting area. So it uses active means to collect the sample. It also has an iris that closes and opens so that the sample is protected after collection. The pilot gets to turn the switch on and off to control that iris. And this is it pulling in that sample from a real dolphin. So we know it works. There's the snot. That's what we're looking for. So we take that, collect that, and do a health assessment on it. So we're going next month. This is the dolphin in Bermuda here. We're going out there. She's going to swim out into the open ocean for us. She's trained to do this. And we're going to chase her down with our PHASM to collect her blow samples. So that's a fun activity next month. And then we'll be using PHASM in Galveston area. So if you're in Galveston, you see a flying drone near dolphins, that's us. Hi. We'll be doing that over 2023 to 2025. We're also developing systems on the drone that help the pilot fly like this, because this is a very difficult thing to do. So we're building artificial intelligence into the drone so that it knows how to approach the dolphins and how to position itself depending on prevailing winds so that it collects the sample more accurately. So we're using thermal for that, and we're using simulation modeling that goes into the drone's brain to help us figure that stuff out. So I want to thank funders from the European Union, Institute for Citizens and Scholars, University of Chicago, Dolphin Quest, SFA, and OSU for all the funding support for this. And if there are any questions, I'm happy to answer them.
So you were talking about that CD36 receptor.
Yep.
So I guess if you already, well, my first question would be, is the sequence for the genome of bottlenose known?
Yes, bottlenose is a known genome, and what we did is we did some functional genomics with this. So we basically looked at whatever function it serves in the cow, it serves that function in dolphins as well.
Sure. So I guess my question is, has anyone thought to just clone this right into a coli and then just do NMR on that protein purified from coli?
That's a really good question. Now, the only things that we've done in terms of protein purification is we looked at the urine for individualized protein markers to see if there was anything individualized with proteins between dolphin urine cues. That we didn't find anything with. Now, on the aspects that you're talking about with essentially purification models to rebuild CD36, CD36 was discovered in 2006.
Okay.
And so this is very, very new. There's probably 4 researchers in the world who are really working on it.
Oh, well, sure, but I mean, either way, I mean, any cloner worth their weight in dog shit could still even be able to just easily clone it right into E. coli and then, you know,
Could
just
be.
do NMR on it. Sorry, my other question would be, so you said that it, on the screen back where you showed the model of it, or the impromptu model of it, you pointed out glycosylation sites on there. If you're concerned about it, or I shouldn't say concerned about it, but interested in it from a lipid signaling standpoint, why would you not be looking at potentially palmitylation sites on there versus glycosylation?
Those are good questions, and that's why this is a bigger project than just me.
Yeah, fair enough.
So generally speaking, we've been looking at potential collaborators to help us add understanding to this very new thing. So we put this paper out in 2022, literally last year. And so now the next steps are to figure out a lot of that stuff. So this is very new, very, very first of its kind of stuff. By the way, the sense of our body that we know the least about is taste. When I teach this in my physiology classes, the textbook literally has question marks on the diagrams for different aspects of your taste abilities. We don't even know exactly how you taste umami. We don't know. So there's a lot of unknowns here. And hopefully this research will help us start to illuminate in comparative physiology how even we taste certain things. So yeah.
I feel like this entire thing has established like a lot of firsts for us to start learning about bottlenose dolphins, like a lot of information. Like, field of vision is something that probably hasn't done before, and all this other stuff like in general discovering how they're probably the second smartest animal in planets. All this other
Okay.
stuff. So I just want to wonder: is there like any? Is this like the only? Are there other animals or other, I guess, research departments that are doing this kind of stuff with other animals?
I will tell you this, folks. I can count the number of researchers who work on bottlenose dolphins and on these systems with both of my hands. We all think there's like thousands of dolphin scientists out there that are doing a lot of work. That's not true. In fact, it's very hard to get into doing some of this dolphin work because dolphins sometimes are hard to access, especially in the wild. So that's why we do a lot of work with zoos and aquariums. So no, there's not a ton of scientists out there doing this. So that means that there's wide open areas to study. I will tell you, animal biology, studying new things in biology, it's easier than you think to discover new things if you're well enough read. If you read enough research and know what is known and unknown about your field, discovering new things is not as hard as you think it is. It's just about knowing what we know and being creative enough to find new ways of getting at certain questions. But yeah, look, There's a lot of wide open road here for people who are interested in this kind of stuff. I assure you there's plenty of room for all of us.
When it came to the differences between figuring out the social patterns, or I guess signature whistles, wild and more domestic dolphins, what are some differences that really stood out to you and your team?
So because dolphins innovate their whistles, you'd think, oh, well, maybe they've got accents, or maybe they've got different types of vocalizations depending on the different areas they're in. There's one group of researchers that thinks they found differences in signature whistles based on region, based upon where the animals live. So if they have like a sandy bottom versus a hard bottom on the ground under the water, then they'll have a different signature whistle that'll accommodate for how sound would bounce off those two things. I need to see more data on that to believe it, because dolphins are just innovating like crazy. And like, I don't think they're playing that kind of fourth dimensional chess about sound dynamics. They could be, but I just want to see more evidence about that. Right now, I know that dolphins innovating signature whistles the way that they do. One of the things that we do know, for example, Nolani had that big flat part in her whistle. Remember that kind of like flat whistle? That whistle's flat at 10 kilohertz. 10 kilohertz is the same frequency as a trainer's whistle when the dolphin does a good thing. So basically what she did is she took part of the human's whistles and made it part of her signature. She's also a very human-centric dolphin. Anytime you walk around the thing, Nolani's the one that follows you wherever you go. She's very human-focused. So we know that human-focused dolphins tend to bring in parts of that 10 kilohertz whistle into their signature whistles. So we know how all that learning and all that kind of social interaction plays the predominant role in the development of the signature. So it's more, I don't think it's more regional. I think it's more, who's this dolphin hanging out with, and which elements does it just happen to like as it pulls into its whistle.
So what's known about the dolphin hippocampus?
It is a little smaller per body size than us, a little smoother per body size than us. Sam Ridgway has done this work. And so if you want to get a handle from his original works, that's where to find it. Generally speaking, however, we shouldn't assume too much about that. Only because we know that in dolphins, a lot of things that we assign in certain areas of our brain are reassigned in different areas in theirs. So the functional neuroanatomy, if you think, by the way, if you think we know very little about their sensory capabilities, we know even less about functional neuroanatomy in dolphins. We just don't have the easy technology to put electronics on them and get these data as they're doing things in salt water. So it's always been very tricky. But that I can tell you is the basics of what we understand about the hippocampus, only from dissection, not from functional neuroanatomy. But good question, yeah.
So this is just something I don't fully understand. You were saying the reason that people were skeptical about the whistles was they weren't sure if they were representative
Yes.
of individuals. So how is the urine samples different? How can you know for sure that those are also not, or those are certainly representative?
Okay. Well, that's the one thing a dolphin can't fake. It's an honest indicator of who they are, is their urine cue. And that's the funny thing about dolphins. They can take each other's whistles if they wanted to. They don't actually. When a dolphin refers to another dolphin, what they do is they make a mistake. And we call it an intentional mistake, even if we don't know if it's intentional. But they basically, if the whistle is this, the difference when they do the other dolphin's whistle to call them over will be to put a little flare at the end. Okay? So that's how a dolphin refers to another dolphin. And they'll only do one bout of it. They won't do the repeated bouts. So that's how you know that the dolphin is essentially not trying to commit identity theft. But urine, you can't do that with. So the urine paired with the whistle is basically the dolphin telling us, I know which signature whistles go with which individual, and that individual by chemical is locked. And so that's really the trick to it, yeah.
Thanks for watching. Please like, comment, and subscribe for more content from LSC-Kingwood.